earlier lecture, that is Chemical Foundation of Physiology Part 1, we have learnt about solution, osmotic pressure and diffusion. Now, in this lecture, you are going to learn about some other very important physiological processes like pK, pH and buffers, where pK is acid dissociation constant, pH is potential of hydrogen ion and buffer is a property of solution to keep check on pH. Let us try to understand each one in considerable detail, that how they are responsible for controlling various physiological functions of the body.
pK and pH. An acid dissociation constant pKa, also known as acidity constant or acid ionization constant, is a quantitative measure of the strength of an acid in solution. It is the equilibrium constant for a chemical reaction known as dissociation in the context of acid base reactions. The equilibrium can be written symbolically as Ha give rise A minus, that is acetate ion plus hydrogen ion. It is a balance reaction, where Ha is a generic acid that dissociates by splitting into acetate ion, known as the conjugate base of the acid, and the hydrogen ion or proton H plus, which in the case of aqueous solutions exist as a solvated hydronium ion. In the example shown in the figure, Ha represents acetic acid and A minus the acetate ion. The chemical species Ha, A minus and H plus are said to be in equilibrium when their concentrations do not change with the passing of time. The dissociation constant is usually written as a quotient of the equilibrium concentrations in mole per liter, denoted by concentrations of Ha, that is concentrations of acid, then concentrations of acetate ion and the concentrations of hydrogen ion. So, the dissociation constant Ka is equal to concentrations of acetate ion into concentrations of hydrogen ion upon concentrations of acid. What is weak and strong electrolytes? Strong electrolytes are almost completely ionized in aqueous solution, whereas weak electrolytes are only partially ionized and a solution contains the undissociated compound as well as ions. Many acids are weak electrolytes and partially dissociate to produce hydrogen ions and the generalized anions. It follows then that a measurement of the hydrogen ion concentration of a solution of a weak acid does not give the total hydrogen ion concentration, because some of the hydrogen remains in the undissociated form. However, if the acid is titrated, the undissociated compounds progressively dissociates and eventually the total hydrogen ion concentration is found. This is why the titrable acid is distinguished from the actual acidity, what we call the pH. With strong acids, the titrable acid and the acidity are the same. Derivation of pK. A weak acid dissociates in reversibly in water, that is generic acid Ha, it dissociates into hydrogen ion and acetate ion. The dissociation constant K defines the ratio of the concentrations of the dissociated ion and the undissociated acid. Square brackets denote concentration. Dissociation constant K is equal to concentrations of hydrogen ion to concentrations of acetate ion upon concentrations of acid. Rearranging the equation to isolate the hydrogen ion concentration, that is concentration of hydrogen ion is equal to dissociation constant K into generic acid, that is concentrations of Ha upon concentrations of acetate ion. Convert both sides of the equation to log base 10. becomes logarithm of hydrogen ion concentrations is equal to log K plus log concentrations of acid minus log concentrations of acetate ion. Multiply by -1 to create the term minus log hydrogen ion concentrations. So, it becomes minus log hydrogen ion concentration is equal to minus log K minus log acid concentration plus log acetate ion concentration. Substitute the term pH for minus log hydrogen ion concentration and pK for minus log K. So, it becomes pH is equal to pK plus log acetate ion concentration upon acid concentration. So, now pK is equal to pH plus log acetate ion concentration upon acid concentration. The end results are two terms. One is pH, the familiar measure of acidity or alkalinity, the other is pK. What is pK now? Actually, it is a very useful term. One way of looking at its value is to consider the phenomenon of pH buffering. Buffer action is a property of a weak acid and its salt. Unlike the weak acid, the salt is fully dissociated. If we assume that the pK of the weak acid is 5, which are an entirely plausible value, then a concentration of acetate ion due to the presence of the acid is 0.00005 mole for each mole of acid. This means in effect that the acid's contribution to the total acetate ion concentration is overwhelmed by the salt. So, it is possible to write the equation opposite as a good approximation which ignores the contribution to acetate ion concentration by the weak acid. The Henderson-Hasselbalch equation goes like this: pH is equal to pK plus log concentration of salt upon concentration of acid. Let us summarize it. The dissociation constant K defines the ratio of the concentrations of the dissociated ions and the undissociated acid. The Henderson-Hasselbalch equation relates pK, pH and the ratio of salt concentration to undissociated acid. The buffering action of any weak acid salt mixture is at its greatest close to the pK. Let us look at pH. Let us know the basics of acid and base. When inorganic acids or bases dissolve in water, they dissociates. They is then separate into ions and becomes surrounded by molecules. An acid is a substance that dissociates into one or more hydrogen ions and one or more anions. Because hydrogen ion has one positive charge, an acid is also referred to as a proton donor. A base, by contrast, removes hydrogen ion from a solution and is therefore a proton acceptor. Many bases dissociate into one or more hydroxide ion and one or more cations. To maintain homeostasis, intracellular and extracellular fluids must contain almost balanced quantities of acid and bases. The more hydrogen ion dissolved in a solution, the more acidic the solution. The more hydroxide ion, the more basic solution. The chemical reactions that takes place in the body are very sensitive to even small changes in the acidity or alkalinity of the body fluids in which they occur. Any fluctuations from narrow limits of normal hydrogen ion concentration and hydroxyl ion concentrations greatly disrupts body functions. A solution's acidity or alkalinity is expressed on the pH scale, which extends from 0 to 14. This scale is based on the concentration of hydrogen ion in moles per liter. The scale for measurement of pH was developed taking water as the standard. A pH of 7 means that a solution contains 1/10 millionth, that is 0.0000001 of a mole of hydrogen ions per liter. The number 0.0000001 is written as 1 into 10 raised to minus 7 in scientific notation, which indicates that the number is 1 with the decimal point moved 7 places to the left. To convert this value to pH, the negative exponent minus 7 is changed to a positive number 7. For example, a solution with an hydrogen ion concentration of 0.0001, that is 10 raised to minus 4 moles per liter, has a pH of 4. A change of one whole number on the pH scale represents a 10-fold change in the number of hydrogen ion. For example, a pH of 6 denotes 10 times more hydrogen ion than a pH of 7. The midpoint of the pH scale is 7, where the concentrations of hydrogen ion and the hydroxyl ions are equal. A substance with a pH of 7, such as pure water, is neutral. A solution that has more hydrogen ion than hydroxyl ion is an acidic solution and has a pH below 7. A solution that has more hydroxyl ion than hydrogen ion is a basic, what we call an alkaline solution, and has a pH above 7. Buffers. Buffers are chemical substances which are able to bind free hydrogen ion or hydroxyl ion without change in pH. Although the pH of body fluids may differ, the normal limits for each fluids are quite narrow. Table shows the pH values for certain body fluids. Homeostatic mechanisms maintain the pH of blood between 7.35 and 7.45, which is slightly more basic than pure water. If the pH of blood falls below 7.35, a condition called acidosis occurs, and if the pH rises above 7.45, it results in a condition called alkalosis. Both conditions can seriously compromise homeostasis. Even though strong acids and bases are continually taken into and formed by the body, the pH of fluids inside and outside cells remain almost constant. One important reason is the presence of buffer systems, which functions to convert a strong acids or bases into a weak acids or bases. The principal buffer system of the body fluids are the protein buffer system, the carbonic acid bicarbonate buffer system, and the phosphate buffer system. A certain amount of all three is normally present in the body and cellular fluids, and so the maintenance of a constant pH depends on a complex system. Let us say protein buffer system. The protein buffer system is the most common buffer in intracellular fluid and blood plasma. For example, the protein hemoglobin is an especially good buffer within red blood cells, and albumin is the main protein buffer in blood plasma. Amino acids, the components of protein, contain minimum one carboxyl group COOH and at least one amino group NH2. These groups are functional components of the protein buffer system. The free carboxyl group at one end of a protein acts like an acid by releasing hydrogen ion when pH rises. It dissociates as follows. The hydrogen ion is then able to react with any excess hydroxyl ion in the solution to form water. The free amino group at the other end of a protein can act as a base by combining with hydrogen ion when pH falls as follows. So, protein can buffer both acids and bases. In addition to the terminal carboxyl and amino groups, side chains that can buffer hydrogen ion are present on 7 of the 20 amino acids. Carbonic acid bicarbonate buffer system. The carbonic acid bicarbonate buffer system is based on the bicarbonate ion, that is SCO3 minus, which can act as a weak base, and carbonic acid H2CO3, which can act as a weak acid. If there is an excess of hydrogen ion, the bicarbonate ion can function as a weak base and remove the excess hydrogen ion as follows. The carbonic acid H2CO3 dissociates into water and carbon dioxide, and the CO2 is exhaled from lungs. Conversely, if there is a shortage of hydrogen ion, the carbonic acid H2CO3 can function as a weak acid and provide hydrogen ion as follows. Phosphate buffer system. The phosphate buffer system acts via a mechanism similar to the one for the carbonic acid bicarbonate buffer system. The components of the phosphate buffer system are the ions dihydrogen phosphate H2PO4- and monohydrogen phosphate HPO4-2. Phosphates are major anions in intracellular fluid and minor ones in extracellular fluids. The dihydrogen phosphate ion acts as a weak acid and is capable of buffering strong base such as hydroxyl ions as follows. The monohydrogen phosphate ion is capable of buffering the hydrogen ion released by a strong acid such as hydrochloric acid by acting as a weak base. Because the concentration of phosphate is highest in intracellular fluid, the phosphate buffer system is an important regulator of pH in the cytosol.
Thus, acidity constant is significant to understand several chemical reactions taking place in the body. When some chemical compound gives or accepts proton, its pH change and act as acid, base or neutral entity. So, there exists different buffer system in the body to keep the pH value at desired level and maintain homeostasis. Hence,